So we're going to go through a little bit of information about the actual impulse now. So we've previously looked at action potentials, which is where we reach around plus 30 millivolts inside the axon. But the impulse, which is what this lesson is about, is how does that then travel along the axon? We call that movement along it being propagated. Okay, so if we talk about an impulse being propagated along a neuron or an axon, it means that charge, the plus 30 millivolts, moving along the axon. Okay, so you have the pages down here in case you need them to help you as well. So impulses. So propagated, like I said, just means the action potential actually moving along the axon. Now, what's actually moving along the axon is sodium ions that are diffusing away from the site of the action potential. So, if you imagine that the sodium ions diffused through the voltage gated channel proteins into the axoplasm, they're going to be in a very high concentration in the axoplasm, and they're going to start to move from a high concentration to a low concentration, and that's going to be in both directions, so forwards and backwards from where they've gone in. Okay. Now, this movement of ions, if you think a movement of a charge, now that's a current. Okay, so a key word we have to use when we're describing what happens is as the ions diffuse, they create localized currents. We'll have a look at what that looks like on the axon in a moment. Now, that localized current is just the ions moving through the axoplasm. And as the ions diffuse, what they do is they take that positive charge with them and they depolarize the adjacent or neighboring region. Okay, so that adjoining region, the region next to the original one where they diffuse through the membrane into the axoplasm, they start to depolarize the next region along the axon. And as we know, as that depolarizes, sodium ion voltage gated channel proteins would start to open. Okay, now if they open in the adjoining region, sodium ions move through into the adjoining region. They then again diffuse to the exoplasm. They create local currents, okay, and then that will depolarize the next adjoining region. Voltage gated channel proteins open and sodium ions diffuse in there. So it's a bit like a Mexican wave. Now, the region that originally had the sodium ions diffusing in, once they've actually moved in and you've reached an action potential, which is plus 30 millivolts, those sodium ion voltage gated channel proteins would actually close. And this part of the axon will go through what's called a refractory period. Now, that means the sodium ion voltage gated channel proteins are inactive for a period of time. And the whole point of this is to create individual impulses. Okay, so it means that you don't just have a one constant impulse going to your brain, because what your brain can actually tell by the information it receives is how many impulses there are, for example, a second. Now that gives the brain information. If it was one constant impulse, the brain wouldn't be able to tell anything from that. So the refractory period is really important. So after an action potential, a second action potential can't be initiated. And that's because once those sodium ion voltage gated channel proteins have opened and the sodium ions have diffused in and you've reached plus 30 millivolts, the sodium ion voltage gated channel proteins become inactive until we get back to the resting potential of the axon. So this is the refractory period. We'll look at it on a graph in a moment. So here we have an image of the neuron. This is a motor neuron. Okay, we can tell because of the large cell body, this end with the nucleus. Here is the axon. Now, if we look at the axon, this whole red section is actually still in resting potential. It'll be around minus 70 millivolts inside with a more positive charge outside. If you remember, it's positive on the outside because a lot of sodium ions have been built up on the outside compared to inside. And there's a lot of potassium ions on the inside. And there's also chloride ions that help to make it negative and cations as well, so negatively charged proteins. Okay, so some stimuli would occur here at the beginning of the axon. We learned about this part last lesson. That stimuli would cause some sodium ion voltage gated channel proteins to open. As you can see, the sodium ions would diffuse in down the electrochemical gradient and they move into the axoplasm. And you can see they're actually going to then diffuse from a high concentration here to a lower concentration to the right and to the left. Now, because they move to lower concentration to the right, they actually create here in the next section that localized current that we talked about because they're taking their charge with them, remember? And that localized current causes in the next section of the adjoining region the sodium ion voltage gated channel proteins to open. Sodium ions will diffuse in. If we hit threshold again, then more sodium ion voltage gated channel proteins open and more sodium ions diffuse in, and we'll get an action potential of plus 30 millivolts. Now, those sodium ions again will diffuse from high to low concentration to the right and to the left. They'll create a localized current, but the only side they're actually going to affect, if you look at it, is the side to the right. Okay, so the side in front of that previous action potential. If a localized current is created here and here, that means some sodium ion voltage gated channel proteins will open and the sodium ions will move into this section. Sodium ions are diffusing back as well, but if you remember then from what we just said, this section that already had an action potential is in the refractory period, which means the sodium ion voltage gated channel proteins are currently inactive. They cannot open. So another important thing about this refractory period, other than allowing discrete impulses, it also means that the action potential can only be propagated in one direction along the axon, which is very important. We don't want it going back again. We need it to go towards the axon terminals. So it's a bit like a Mexican wave, isn't it? The same thing has happened here then. We had our localized current. Localized current causes some sodium ion voltage gated channel proteins to open. Sodium ions diffuse into the exoplasm. More sodium ion voltage gated channel proteins will open if you reach threshold, will reach plus 30 millivolts, and those sodium ions will diffuse to the right and the left again, creating localized currents again here on the right. So each tiny section of the axon has to be depolarized. And we can see again the previous section is in the refractory period, and the section here that was in the refractory period has now returned to resting potential again. So that means here another action potential could then start. So we can see here, in this way, each little section of the axon has to become depolarized in order to pass that action potential along and propagate it all the way along the axon. Now, there's a way of speeding this up, and that's called saltatory conduction. Okay, so saltatory conduction is particularly seen in vertebrates, okay, where we have axons insulated with myelin sheath. You might remember this from before. Myelin sheath is made up of Schwann cells, which are wrapped around the axon, and they basically act as an insulator. So what that means is, being an insulator, they prevent ions from moving in or out of the axon wherever the Schwann cells are present. Ions can therefore only move in the gaps between the Schwann cells, which are called the nodes of Ranvier. Okay, so any action potential that's reaching plus 30 millivolts would only happen in a node of Ranvier because that's the only place the sodium ions can move. And this creates what we call a jumping action potential. Okay, now this jumping style of action potential is what is called saltatory conduction, and it's much faster. So if you look here, you've got the Schwann cell wrapped around this axon. So no sodium ions or potassium ions can move in or out of the axon where that Schwann cell is present because it's insulating. They can only move here. Now that's got quite a few advantages. First of all, it's actually helpful for the axon to maintain its resting potential because normally some potassium and sodium ions would actually leak through the channel proteins, and the sodium potassium pump would have to keep moving them back into place to maintain the resting potential. But because more of the axon is covered in myelin sheath, less of that leakage occurs. And actually, if you think you've only got sodium potassium pumps in each of these nodes of Ranvier, they're not going to be any present in any other sections. That means you're using far less energy in order to actively transport those ions to the appropriate place. If you're thinking about the voltage-gated channel proteins and the other channel proteins, they are also only present in the nodes of Ranvier. And the impulse would go like this. It would appear in this node, and then we say it's jumping. It doesn't actually jump, but then it would appear here, and it would appear here, and it would appear here, and so on. And you can see how it looks like it's jumping, but we'll explain what it's actually doing now. So, if we have a look at this axon, you can see the jumping saltatory conduction is represented by the arrows. What's happening here is exactly the same as happens in the unmyelinated axon. Okay, so for example, if we take this as an area where an action potential has happened here, the sodium ions have moved in. They're going to diffuse again to the right and the left through the axoplasm. But this time, they diffuse through the axoplasm. And the only place they create a localized current is actually all the way across this Schwann cell. Okay, so we get a localized current affecting the next adjoining region at the next node of Ranvier. It doesn't have to depolarize this section and then this section and then this section and then this section and so on, which is what takes so long. It can skip out that whole section with the Schwann cell because it's insulated, the ions can't move out. They diffuse through the axoplasm, you get a localized current. That localized current affects the next node of Ranvier, and the node of Ranvier will be where some of the sodium ion voltage gated channel proteins open, sodium ions diffuse in, and again more will open. And then that happens again with the next node of Ranvier, so it's much faster. You can see here, sodium ions are diffusing in through the voltage gated channel proteins. They diffuse right and left, and you get the localized current here. They can't move out because the Schwann cell, remember, insulates the axon, so they're not going to leak out. They just diffuse through, create a localized current for the next node, and then we'll get an action potential in the next node as well. We won't get one in the previous node because that will be in the refractory period. Okay, so saltatory conduction requires less energy, okay? This is meant to say axon, sorry about that, okay? And that's because active transport of the ions only happens at the nodes, like we said. So there's less sodium potassium pumps. The ions are less likely to leak out because of the insulation, and therefore less respiration is needed, less ATP is needed in order to maintain the ions in the correct positions. Now, temperature can affect the movement of the ions in both situations. If you think about it, higher temperature means more kinetic energy, and therefore ions are more likely to move faster. Okay, and it can actually increase the conduction of the impulse. However, above around 40 degrees Celsius inside your body, it would actually be quite dangerous, okay, because enzymes and proteins would change shape and denature. Up until 40 degrees, it's actually beneficial because the enzyme rate of reactions would occur faster. So things like aerobic respiration would release more ATP. And also, diffusion would occur faster. So the ions, the sodium ions would diffuse in faster, the potassium ions would diffuse out faster, and therefore it'd be quicker to propagate the impulses, but also to go back to the resting potential afterwards.